No. So when it comes to photosynthesis, we discussed that photosynthesis can have two distinct stages. A light-dependent stage followed by a light-independent stage. So in this particular segment, we will talk about the light-independent stage of photosynthesis. Not the light-dependent, but the light-independent stage. Now, bear in mind that the light-independent stage of photosynthesis is typically known as the Calvin cycle. Known as the Calvin cycle. Typically known as the Calvin cycle. Now, obviously, as the name suggests, this is known as the Calvin cycle because it is indeed a cycle. So, starting off this particular cycle, we have a very important compound. I'll be using this faint blue to represent that important compound. So, this is what we are starting with. 1, 2, 3, 4, 5. Now, technically, as per normal collocations, I will simply be using a circle to represent a carbon atom. So in this case, we have 1, 2, 3, 4, 5 carbons. So whatever compound I'm about to show you has 5 particular distinct carbon atoms to it. And this 5 carbon compound is known as RUBP. Known as RUBP. RUBP stands for ribulose bisphosphate. Ribulose bisphosphate is pronounced just like this. Ribulose bisphosphate. Bisphosphate. Ribulose bisphosphate. RUBP for short. Very simple. RUBP for short. Okay, RUBP for short. This is now, when it comes to the thing with cycles are, you have to find a starting. It's very hard. Sometimes not hard, but it's kind of hard. So for the Kelvin cycle, I recommend you use this as your starting point. Right, so then what happens? This ribulose bisphosphate is converted into two of these. Now, one, pay attention to the carbon number, okay? 1, 2, 3, 1, 2, 3. So now here we have two glycerate phosphates. Here we have two glycerate 3-phosphates. Glycerate 3-phosphates. Now if we pay attention to the carbon number, we can see it's 1, 2, 3, 1, 2, 3, a total of 6 carbons. Now how can we possibly form a 6 carbon compound? Now the actual thing is, here's the deal. We form a 6 carbon compound, but this thing is very unstable. So that's why it breaks down into two threes. Two threes. Okay, that's the thing. So then for us to go from 5 carbons to a total of 6 carbons, in some form of way, a carbon has to be added. In some form of way, a carbon has to be added. So how does that happen? That happens by adding a carbon dioxide. Carbon dioxide is clearly a one carbon molecule. It's clearly a one carbon molecule. So how exactly now? Carbon dioxide is available in air. Agreed. We understand that. But how or what enzyme or metabolic pathway or organism actually introduces this carbon dioxide into the Calvin cycle? That's our question. Who introduces this carbon dioxide? That is done by an enzyme known as rubisco. Rubisco. The rubisco enzyme fixes. We call it that we fix. You know, in the nitrogen cycle, we have nitrogen fixation. So in this case, we have carbon dioxide fixation. Rubisco fixes carbon dioxide. That's what it does. Okay. So then what happens? Rubisco fixes carbon dioxide, and then we have a 1, 2, 3, 4, 5, 6. This is actually an unstable 6 carbon intermediate. Knowledge of this step is not required for A2 levels, but I just mentioned it, so now I'll scratch it. So simply a 6 carbon unstable intermediate is formed, so therefore it's just going to break into 2 GPs. That's the short form. Glycerate phosphate is GP. 2 glycerate phosphates. Simple, right? And then we have the final substrate of this. I mean, final reaction metabolic pathway component or compound or constituent. That would be, once again, so 1, 2, 3, 1, 2, 3. Now we have 2 triose phosphates. 2 triose phosphates. So TP for short. TP for short. We have 2 triose phosphates. Let me adjust this arrow a little bit. Okay, we have 2 triose phosphates, like so. And then we're going from the triose phosphate back to the RUBP, ribulose biphosphate. The cycle is now complete. Now let's just look at the metabolic pathway and identify anything's produced, anything that are added, anything that are removed. Anything that are removed. Nice. Okay. Now, right over here, during this process of conversion of this glycerate phosphates to triose phosphates, we have ATP being used. ATP is not being produced. Okay, now if we recall from our previous non-cyclic and cyclic photophosphorylations, ATP is carried on to the Calvin cycle. Remember, ATP is carried on to the Calvin cycle. Now, in the case of non-cyclic photophosphorylation, ATP and NADPH is carried on to the Calvin cycle. Yes. In the case of cyclic photophosphorylation, only ATP is carried on to the Calvin cycle. Okay, so here we have ATP being carried on to this particular cycle. Okay, that's a fact. That's something that we know. Now, actually, this is why that ATP is brought here. That ATP is now utilized. So it's broken down. When ATP breaks down, we once again get adenosine diphosphate and an inorganic phosphate, ADP plus PI. So now do you see this process requires energy. Conversion of GP to TP requires energy. We have to use a ATP molecule. ATP molecule. At the same time, so one of those NADPHs that we carried on from the photophosphorylation sites are used, and it becomes NADP plus. We are using everything here. We are using an ATP, we are using the NADPH to convert glycerate phosphate into triose phosphates. Okay, into triose phosphates. Triose phosphates. And one final little thing, when we are converting PP back to RUBP, we need some energy too. So one final ATP utilization to produce ADP and inorganic phosphate, like so. Inorganic phosphate, like so. So that is the basic outline. This is all the information that you need for the Calvin cycle. Now I would like to add some excess information onto this so that questions become easier and you can answer certain pathway questions more easily. And after that, I'll give you a very shortened version of this. This is the completely packed and compact, non-compact and all iterated type of thing. No? after this, I'll give you a very shortened form of this so that it's easier to remember. Right, hold on. Let me pick something like a yellow color. Okay, right. So now, these glycerate phosphates that are produced, these glycerate phosphates, hold on, before that I will actually include, like I'll actually show that we are bringing NADPH, and ATP from the phosphorylations. So we are bringing those things and thus they are being utilized for the Calvin cycle. Okay, right, back to what we were talking about. So when it comes to this glycerate 3-phosphate, this is the most usable intermediate in this pathway. What is this? This is the most usable intermediate in this particular pathway. This glycerate phosphate is converted into carbohydrates like starch, cellulose, and even sucrose. And that glycerate phosphate can also be converted into amino acids and also lipids. This glycerate phosphate, GP, is the most used intermediate, and that is used to produce carbohydrates, amino acids, and even lipids. Not just that, the triose phosphate is also useful to produce these. So I'm just gonna draw an arrow onto here just like that, onto here just like that. Now, if you have noticed, I don't know if you noticed, but if you did, you can see that here we have 6 carbons. I mean, it's broken down into 2 because it's unstable again, but here we have 6 total carbons. Converting back to ribulose bisphosphate, we have only 5 carbons now. So that would either mean that we have lost the carbon dioxide or something else has happened. Now, in the Calvin cycle, we don't lose carbon dioxide. We obtain carbon dioxide into the cycle. So there's no losing step. So then what on earth happened to this extra carbon? What happened to that carbon? Thing is, when this pathway occurs, 5 out of 6 parts of the triose phosphate molecule is converted into ribulose bisphosphate. And 1/6 of the molecule is utilized for this, for the production of organic and inorganic substances. Okay, do you get that? So this is 6 carbons now. So 5 carbons are used back to produce ribulose bisphosphate. 1 carbon is used to produce carbohydrates, amino acids, and lipids, and all of that. Okay, so if I zoom out and show you the entire process, like so, this will be, let's get rid of that. This will be the entire Calvin cycle with all the A2 level information that you have to know. Now let us outline the very shortened form of this. Very simple. Pay attention. So we obviously know we start off with RUBP. What is RUBP short for? Ribulose biphosphate. Converted to 2 times GP. Glycerate phosphates. Converted to 2 times TP. Triose phosphates and both. That's the first step you should do. Just write the intermediates and complete the cycle. Then you can fill in every other thing. We know carbon dioxide is fixed in by an enzyme called Rubisco, like so. An enzyme called Rubisco. Simple. We know all the things that we brought from the photophosphorylation stages are being utilized. So we have ATP, we have NADPH being converted back to NADP plus, and then here we have a final ATP being utilized, ADP plus inorganic phosphate, like so. That's it. That is it. Okay. This is a completely informative cycle with everything you need to know. This is a very shortened form of the cycle so that you can remember better in exams. Please do remember that GP is used to um, make all of the carbohydrates and everything and all that stuff. And 1/6 of this molecule is also used for those purposes. Keep that in mind as well. That's not included in the shortened form. So all in all, overall, that would be the light independent stage of photosynthesis in a nutshell. With, of course, all the information that you have to know.